Okay, welcome back. So we're going to pick up in this part talking about how we are able to modify our pain perception. So we've talked about different types of sensory receptors. We've talked about different types of sensory modalities, meaning the different types of stimuli that those receptors can respond to. And one of those receptors was a nociceptor, right? So if you recall, a nociceptor was a sensory receptor responding to pain, right? And so we said that pain was a very good thing generally, a very protective, very healthy, very physiological response. But at some points, and sometimes we want to modify our perception of it, right? So we want to modify how much we are actually feeling that pain, right? So we can do this via a couple of different mechanisms. So one of these is something known as just an analgesic. And so this is modifying our perception of pain. An example of this is an NSAID. So this means a non-steroidal anti-inflammatory drug. And so these are things that you know as Advil and Aleve, right? So these are medications that can decrease the synthesis of different molecules and peptides and proteins that can then stimulate the nociceptors. And then if you stimulate the nociceptors, you get the pain. So if you block those chemicals and peptides and proteins from stimulating the nociceptor, then you block that stimulation, so you block the formation of an action potential, and so you block your perception of that pain, or you really block the generation of the action potential that's leading to that pain, right? So NSAIDs are one effective way, one good way at preventing our pain sensation and perception. There are obviously side effects associated with every medication, and so for Advil and some other associated NSAIDs, one common side effect is you can have gastric ulcers. You can develop gastrointestinal problems. This is because there's something known as prostaglandins, and these prostaglandins are essentially little proteins, little messengers that can stimulate our nociceptors that then lead to the formation of an action potential and lead to the perception of pain. There are also prostaglandins that lead and contribute to the lining of our stomach that coat it and protect it from the acidic environment of the acid in the stomach. Well, if you are taking medications that block the formation of prostaglandins, they can also block that formation of the gastrointestinal lining, and so you can have acid then come and irritate the inside of the stomach, and you can get these gastric ulcers. Okay, so long term, and if you're taking these medications just briefly, like just once every couple of months as you need pain or something, there's nothing to worry about. But if you're, generally, there's nothing to worry about regarding those long-term side effects. But if you're taking them very chronically, very consistently, all the time for a long period of time, then yes, you can start to run the risk of having some serious long-term side effects as a result of taking those medications. So there's all these other medications as well. There's anesthetics that we'll talk about in a second. There are opioids. Both endogenous and synthetic opioids, and we'll talk about those. And then there's things like anticonvulsants, good for neuropathic pain. We won't talk about that too much, but we talked about phantom limb pain previously in the previous part. And these anticonvulsants or these anti-epileptic medications can be really good at treating that neuropathic pain, meaning neuropathic coming from the actual neuron, that kind of sharp burning pain that's associated with certain types of pain. So we also have anesthetics, as we mentioned. So anesthetics are blocking the formation of an action potential, okay? So we know that we have some stimulus, right? We have a temperature or a chemical or something that's resulting in the depolarization of a cell. And so you get an action potential, and if you get an action potential, you get that impulse traveling towards the nervous system, toward the central nervous system, and then to the brain, and you perceive that as pain. Well, one way to prevent that perception of pain is to block the formation of the action potential. And so there are medications like lidocaine and tetracaine and novocaine. These can bind and block sodium channels, right? These voltage-gated sodium channels that open in response to a voltage or in response to some stimulus. And so here on the left, you can see you have sodium flow in, right? You have, just like we talked about with excitable cells, sodium flows in. You depolarize the cell, so you get up to threshold, you have depolarization, you have repolarization, and all this. Well, on the right here, now you have your medication. You have lidocaine, bupivacaine, you have tetracaine, lidocaine, novocaine, whatever it may be. If you block that entry for sodium, that route where sodium could then enter the cell, then you block the depolarization. So here, you see you have a little bit of depolarization, but you probably don't reach threshold, so you don't get the full action potential. Well, if you don't reach threshold and you don't get an action potential, you don't get that signal traveling to the brain, so there's no way that you could perceive that as pain, right? So anesthetics are a really good way of blocking that action potential generation in sensory neurons, so those afferent fibers or afferent neurons, and that also prevents us from being able to perceive the pain. Now, there are also opioids, right? So we've all heard of opioids. There's an opioid, real problem in the country at this point. There are two general classes of opioids. There are endogenous and there are exogenous, or what are known as synthetic opioids, right? So what we'll talk about here are what are called endogenous opioids, or opioids that come from within us, right, within our body. And these can be things like endorphins, right? So the term endorphin means endogenous morphine, right? So we've all heard of morphine. You may have gotten morphine after you had your wisdom teeth out or after a surgery or some other operation or procedure. It is blocking receptors that are called mu receptors, okay? Or that's one type. So for morphine, it's blocking these mu receptors. When you block the mu receptor, you block the perception of the pain, right? And so this is antinociception or an analgesia. So you're blocking the pain signal from being perceived by the brain, so you don't really notice it that much. Now, when you block the perception of the pain, you still really have the stimulus that's causing the action potential. You still have the action potentials. They're still reaching the brain, but we don't perceive it as really being that bothersome. We don't perceive it as being annoying. We don't perceive it as really being this painful thing, right? And so it's a little bit different than some of the other types of modifiers of pain, okay? Now, one big thing about opioids in general is that they cause lots of different side effects. Some of these are constipation is a very common one, nausea. Another big one is sedation, right? So CNS depression. So this is one big thing you look at in a clinical setting for those taking opioids and those who have potentially maybe been given too much, or those who may have taken synthetic opioids as using it as a illicit drug. And this is one way you can tell if they have potentially overdosed. Okay, there are treatments for that. So there are reversal agents. So things like one is called naloxone, sometimes referred to as Narcan, that can reverse the effects of opioids and of morphine. Okay, so we'll talk here about a little bit about how we can have varying intensities of stimuli, and really not just varying intensities, but how we can determine that different intensities are of a different stimuli and how we perceive things as being very strong or not as strong. Okay, so it turns out that our perception of pain, or our perception not necessarily of pain, but of any sensory stimulus, is dependent on the intensity of the stimulus, and then that intensity of the stimulus translates to more or less action potentials. Okay, so what we see in this picture here is we see sometimes we have small stimuli, sometimes we have medium stimuli, sometimes we have very large stimuli. Depending on those stimuli, we have varying receptor potentials and we have varying amounts of action potentials and varying amounts of neurotransmitter release. So we then perceive it as being a small stimulus, a medium stimulus, or a strong stimulus. So if we look here, this first example, we have a very, very small stimulus. So hardly any stimulus. We have a very small receptor potential. You notice we do not reach threshold. So if we do not reach threshold, we can't have an action potential, so there's no neurotransmitter release. So we don't perceive this signal. We don't perceive this stimulus, whatever it may be, okay? If you go to the next one, you have a little bit stronger stimulus, so something slightly larger. You have a slightly larger receptor potential. You have slightly more action potentials, or you have more action potentials, right? Because now we've reached a threshold, so you have action potentials at the first node, and then you have the action potentials traveling down the axon, and so you get some neurotransmitter release. So you would perceive this stimulus as being present, right? You'd feel it a little bit, right? You would perceive it, you'd be conscious of it, you'd be aware of it. Then you could go even further. So we went from small, medium, now we have a very large stimulus. We have a very large receptor potential because of this large stimulus. At the first node, we reach threshold and we have lots and lots of action potentials. And so we have lots and lots of action potentials down the axon. and then we have lots and lots of neurotransmitter release. So we would perceive this as some stimulus, and we would perceive it as being a more intense stimulus than this previous one in the middle. So the way we can tell that, so take for example, if you had your hand out and I hit you on the back of the hand with a spoon, or I hit you on the back of the hand with a hammer, right? Which one's going to hurt more, right? Probably the hammer is going to hurt more, right? So why does the hammer hurt more? Well, you may say the hammer is larger, the hammer is heavier, the hammer is more dense, right? It moves at a greater speed, something like that. So what that means, what you're saying, is that the hammer is providing a stronger stimulus, right? It's heavier, so it comes down with more force, stronger stimulus. That stronger stimulus results in a greater receptor potential, which generates more action potentials, which releases more neurotransmitter. So we perceive that as being a greater stimulus, right? It hurts more than if I hit you on the back of the hand with a spoon, smaller stimulus, fewer action potentials, less neurotransmitter release, okay? So this is this idea of recruitment, right? So the way that we can determine the intensity of different stimuli is based on the stimulus itself and how strong it is, but that stimulus and its strength will recruit or activate more sensory units, right? So we talked about sensory units previously. That's one axon of one neuron with all of its receptors. So if you increase more sensory units, and if you have a stronger stimulus and that generates more action potentials in that sensory unit, if you put those two together, those two things lead to our perception of this is a strong stimulus, this is a medium stimulus, this is a weak stimulus, okay? Now, there's something else called range fractionation, and this is how we determine kind of a progression of different stimulus intensities, okay? So we can also have certain types of receptors or certain numbers of receptors be stimulated at different thresholds, okay? So you may have, if you think of something like noise, or you think of something like some smell, some chemical that you smell. You have very loud noise, you have medium noise, you have quiet noise, you have very quiet noise. You have things that smell very, very strong, pretty strong, kind of moderate, weak, very weak, right? So we have these ranges here. So depending on the receptors that are responding to the stimuli, we can determine kind of this range of different stimulus intensities, right? So over here we see varying firing thresholds for different stimulus intensities. So what this graph is showing is that at a certain intensity, so for A, at this stimulus here, we have however many action potentials, okay? At this one here, we have so many action potentials and so on and so forth. So it's going to vary slightly, and this variation accounts for our ability to determine this is a very, very strong stimulus, this is a moderate stimulus, this is weak. Okay, so these two things, recruitment and range fractionation, kind of go together, all right? So what we will finish with here is talking about two types of somatosensory pathways, okay? So these pathways are going to be bringing sensory information, so these are specifically going to be afferent pathways, bringing different sensory signals, different sensory stimuli back to the brain, all right? So the first one is known as the dorsal column medial lemniscus pathway, okay, sometimes called the posterior column pathway, all right? So if you recall, the medial lemniscus is a section of white matter, so it's a white matter tract within our midbrain, all right, which is part of our brainstem. So what we have is this pathway bringing different types of stimuli. So this could be pressure, touch, fine touch, vibration, proprioception, okay, from the body, okay? So what we have is we have information coming from the periphery, from the upper limb, from the lower limb, from the trunk, into the spinal cord, up the spinal cord, to the brainstem, to the thalamus, to the cerebral cortex, all right? So just like we talked about previously with the pain pathway, with the spinothalamic, we'll talk about the spinothalamic in the next slide too, you have three neurons, right? You have a primary or a first order, you have a second order, you have a third order. So here we see we have first order here in blue. We're bringing information. So say this is touch information. Say we had, we had the touch information from our hand, okay? So, or our finger. So that information is going to come in via this first order neuron, and it's going to ascend in the spinal cord in a manner that is called ipsilateral ascension. So meaning this signal is going to ascend on the same side of the spinal cord as it came in. So you can see this dotted line running right down the middle. We're staying on the same side. We come in on this side, we stay on this side, and we go up. Okay, we come up to the brainstem, and there are different nuclei. This nucleus gracilis, nucleus cuneatus. These are specific nuclei that are receiving information coming from either the lower limb or the upper limb. So we're saying the upper limb, so specifically that's going to be this cuneatus here. You come in, you go up to the brainstem, and you synapse. Remember, you synapse on a second order neuron. Now, we said earlier with the pain pathway, at some point you have to decussate. At some point you have to cross over, and that's what we see here. We decussate now from this is technically, if we're looking at it here, this may be the left side of the patient, right? So remember, it's from the patient's perspective. So here's the cerebellum back here posterior. So no, this is the left side, and we decussate over to the right side, and we ascend in our second order neuron to the thalamus, right? Part of the diencephalon. We then synapse again on the third order neuron cell body. And then from that cell body of the third order neuron, we ascend in white matter to the cerebral cortex, to a specific region. And again, remember, what part of the brain are we going to? Well, we're going to the postcentral gyrus, and the functional area that's part of the postcentral gyrus is known as the primary somatosensory cortex. Okay, so we ascend, we go up to a specific region. So this, we said, was touch sensation or something coming from our hand or our finger. So it goes to a specific region, whereas if this was coming from the lower limb, it'd go to a different region. All right, so again, first order, up the spinal cord to the second order in the brainstem. We decussate over. We go up on the contralateral side. So now that we've crossed over this midline, we decussated. Now we're ascending contralaterally, meaning on the opposite side. Okay, so we ascend up contralaterally to the thalamus, synapse with third order neuron, go out and synapse in the cerebral cortex. Okay, so hopefully what you get from this is you see the information coming from the periphery is perceived on the opposite side of the brain, right? So here information was maybe coming from the left finger. It ascended and goes to the right side of the brain and is perceived by the right side of the brain, okay? We'll see that some of our motor information happens in the same way. It crosses over and decussates at some point, okay? But that'll be from motor. So this is the dorsal column medial lemniscus. It's called dorsal column because when you look at a cross-section of our spinal cord, there's anterior columns, there's a lateral column, and there's a posterior or dorsal column. The dorsal column is purely afferent information, purely ascending sensory information. And so this dorsal column, we're ascending in that column, and then when we get to the brainstem and the midbrain, we travel within a region known as the medial lemniscus here. We then go up to the thalamus, out to the cerebral cortex, and we are finished there. All right. So this is one pathway. The other one is known as the anterolateral spinothalamic pathway. All right. The anterolateral spinothalamic pathway is bringing information that includes crude touch. So like not very fine touch, but very like firm, crude touch, pressure, pain, and temperature as well. Okay. So the touch and the pressure ascend in what's called the lateral or the anterior pathway, excuse me. And then the lateral pathway transmits the pain and the temperature sensations, okay? So other than that, the pathway is very, very similar, okay? Now, it goes in a slightly different, it doesn't go in the posterior column, but it goes in a region of the kind of anterolateral region. That's why it's called the anterolateral spinothalamic pathway. But the general pathways, the layout's pretty similar. So we have a first order neuron come in, right? Comes into the spinal nerve, to the dorsal root ganglion here, into the dorsal horn, and we synapse, right? What do we synapse onto? We synapse onto a second order neuron. We then decussate, okay? So we have decussated over, meaning we crossed over, and then we ascend contralaterally, right? We ascend in the opposite side of the spinal cord. So we have now ascended up. We've gone through the, so you can see different parts of the spinal cord here, so cervical and lumbar, but we're ascending up through the midbrain, or through the brainstem, medulla, pons, midbrain, and to the thalamus, right? We're now in the thalamus where we synapse with our third order neuron, and from the third order neuron, we go out to our respective parts of the brain, of the cerebral cortex, excuse me. So again, what part of the cerebral cortex? Well, we're going to the postcentral gyrus, specifically the somatosensory cortex. Remember, the postcentral gyrus is part of the parietal lobe, and that somatosensory cortex is the functional area of that gyrus. Now, if you look at this if you look at the third order neurons here in purple, you'll note that one goes to this part of the cerebral cortex, whereas the other goes to this part of the cerebral cortex. So why are they going to different areas, right? So why is one going more medially here, and why is one going more laterally here? Well, it turns out that specific areas of the body are mapped to specific parts of the cerebral cortex, meaning sensory information coming from one area of the body ends up at one part of the brain, and then information from another part of the body ends up at a different part of the brain. So if we follow this up, so this is lateral spinothalamic, so this is from our lumbar region. So this may be a nerve coming from the leg, right? So we follow this up, and it synapses here, and it goes to this region here. So maybe this region here, this is responsible for receiving sensory information from the lower body or the lower limb, whereas if we follow this one from the cervical region all the way up, maybe this is responsible for receiving information from the head and neck area, right? Or the upper part of the upper limb or something like that. So it is mapped very specifically, and this is known as the sensory homunculus, all right? So there is a kind of a strange looking creature, you can look up the sensory homunculus, that stretches over this, this section of cerebral cortex in drawings that shows the distribution of certain parts of the body along different parts of the cerebral cortex. Okay, so in other words, it's mapped. There's also a motor homunculus as well, meaning motor signals, efferent signals going to the fingers arise in a different part of the brain than the motor signals going to the trunk or going to the lower limb or going to the foot. Okay, so it's basically the same thing. Okay, so that is all I have for this lecture. So we covered a lot of information. So review what we have discussed. And again, if you have any questions at all, please do not hesitate to reach out to me, and I'll be happy to answer any questions that you have. All right, I will see you in the next video.